long a time to talk as possible. Many of you know Matt Weitzman. He is a professor of pathology, lab medicine, microbiology, and peds. He's the co-chief of the Division of Protective Immunity at CHOP, and he's the director of the grant review communities at CHOP. He has had incredible amounts of awards and leadership roles and so forth. One of the things that I wanted people to understand today about Matt, who is so honest about his career and the fact that a career is not necessarily from A to B, it could be from A to Z to B, is that the research in his lab has been continuously supported by NIH grants for over 20 years. And they've published over 120 articles in peer-reviewed journals. He is incredibly committed to sponsoring and mentoring young faculty and has created the Grant Proposal Success Program. And Matt is going to tell you a bit about himself, his career, and his commitment to bringing along the next generation. So, Matt.
Okay, can you see that?
Yep, perfect.
Excellent. All right, great. Well, good afternoon, and thank you for coming. I'm really honored to be invited to present in this group. And Lucy and I spoke yesterday, and I decided that I would do something a little different from what I normally do. So if you've seen me speak before, there'll be something new. If you've never seen me speak before, there'll be something new, and I hope you'll all learn something. Okay, so when you start your lab as an assistant professor, you're told really to do two things. One is to focus, and one is to get grants. So when I started my lab, I was completely unfocused, and I probably remain unfocused for most of my career. And I had actually never seen an NIH grant. So I'm going to tell you some of the lessons I learned along the way and tell you a little bit about my journey that got me here. And I entitled this Taking a Lesson from Ben. So probably most of you can guess who the Ben is. This is the Ben. This is Benjamin Franklin, although probably the junior faculty have never seen this image because this is on the $100 bill. And so they're not very frequently given out to junior faculty. So who is Ben? Well, you all know that Benjamin Franklin, that's the name he was given by his mother, has a number of degrees, FRS, FRSA, FRSE. He is described as an American polymath, which basically means he was good at everything. He was a scientist, a writer, an inventor, a statesman, a diplomat, a printer, a publisher, an editor, a philosopher, an intellectual, and much more. He was the founding father of this great country, the United States of America, and he founded many civic organizations, including the idea of lending libraries. And of course, he was the founder of the University of Pennsylvania. So taking a lesson from Matt is what we're here to do today because Ben is not with us. So let me tell you a bit about this Matt. So who am I? So I don't have as many FRS degrees. I have a Bachelor of Science and a PhD. I am not an American polymath, but I am an American and I am a Brit. I share some of these, although nowhere near the level of Benjamin Franklin. So I am a scientist, I am a writer, I am an inventor. I'm not a statesman, I'm not a diplomat, although I try to be diplomatic. I'm not a printer or a publisher, I am an editor, and you can decide whether I'm a philosopher and an intellectual. I am not the founding father of this country, but I am the founding father of the Grant Proposal Success, or GPS program, that we're going to talk about today. And I'm not a founder, but I am a contributor to many civic organizations, including the University of Pennsylvania. So how did I get here? And I'm going to take you back to the very beginning. This is probably the earliest picture of me. On the left, I think this is probably me. This is within the first year of my birth. This was actually the party that celebrated my birth and circumcision, and I shared that with my twin brother. So I think this is my twin brother. Someone needs to mute there. So we were born in a place called Leicester. Leicester, if you don't know it, is in the middle of England. It's on the old Roman route from London to the north to Scotland. If you've heard of it, you may know, if you're a Shakespeare buff, that Richard III was dug up in a car park in Leicester. And if you're a soccer fan, you'll know that Leicester City Football Club one year rose to the top of the league. I then grew up in Cardiff in Wales. If you don't know Cardiff, it's in the capital of Wales. It also has a football team, but it's much more known for its rugby team and for its castle. So I was pushed around by this guy for much of my youth. We were like two peas in a pod. And perhaps that's what got us both interested in genetics. It was really this book about Gregor Mendel that got me interested in the idea of genetics. This is just an amazing story of Gregor Mendel and the discovery of genes and the start of genetics. And this got us both on a similar journey to the point where we actually together edited a book that you can buy about 30-second genetics. It's 50 ideas that come from the field of genetics, with each described in a 30-second description. I went to the University of Leeds. It's an old Victorian university in the north of England. It has some old buildings and then some very modern buildings. This actually has the longest corridor in Europe. I came out of this building. This is where I started to work on viruses. I worked on a bacteriophage, so a virus of bacteria, MS2. And I graduated. There's a picture of me. The sad thing is that I look a lot older than my father looked at my graduation in Leeds. I then went to do a PhD at Oxford Brookes University, where again, I worked on viruses, this time of insects, baculoviruses, which produce a polyhedron coat, and you can replace the gene for polyhedron by other genes, which makes it a really good vector for production of proteins and is used in the pharmaceutical industry. It was also the idea that we were doing there was that we could manipulate this virus to make it a field control agent to control the pine beauty moths of Scotland. So with a Bachelor in Science and a PhD from Leeds and Oxford, I left the UK and headed west. And that's when I came to do a Fogarty Fellowship at the National Institutes of Health in Bethesda, just outside Washington, DC. And after 2 years there, I came to the University of Pennsylvania, where I was a Cystic Fibrosis Foundation Fellowship at Penn. And this sort of summarizes how I think I felt at the time. This is a quote from Mary Shelley's Frankenstein that I really like. None but those who have experienced them can conceive of the enticements of science. In other studies, you go as far as others have gone before you, and there is nothing more to know. But in a scientific pursuit, there is continual food for discovery and wonder. And this wonder and search for discovery was what stimulated me to move west again and set up my own lab. So as a postdoc, I worked on protein DNA interactions of viruses. I worked on this rep protein of AAV and discovered how it targets integration to a region on the human genome on chromosome 19. And then I came to Penn and worked on AAV as a vector for gene delivery, and then the interactions of adenovirus, the helper virus for AAV, to stimulate its replication inside cells. And it was this that started me on a journey to work on virus-host interactions, and I'm still on that journey. So the other thing that I've been interested in from the beginning, also stimulated by the partnerships that I have, is this blending of science and architecture. This is the building where I spent all my time at Penn, the Wistar Institute. At the time, our lab was on the second floor of the Wistar, so I spent a lot of time in this building. And across the quad was this building, which at the time was called the Graduate School of Fine Arts. And my wife was doing her master's in architecture in this building. So they are very, pretty near each other across the quad. This has actually recently been renamed, no relationship, but it's now the Weitzman building. And although these two were very close to each other. We didn't spend much time together. I was in this building, my wife was in this building, until she graduated with her degree in architecture. And she was really intrigued to see one of the most important, I think, architectural buildings in the country. And so we went west to the Salk Institute. So this is a Pennsylvanian, Jonas Salk, looking out on a piece of land that he has acquired in La Jolla, California. And he got together with in a partnership with another Pennsylvanian, Louis Kahn. And together, Jonas Salk and Lou Kahn designed what I think is one of the most impressive and important scientific and architectural buildings in the country, the Salk Institute. So I was honored to be a faculty member of the Salk Institute. It overlooks the Pacific Ocean. Our lab was actually below this level, below the main quad. It's really an amazing, beautiful institute, and also an amazing place to do science. If you're a fan of Kahn, you can see how this building really has all the features of Kahn. It pulls in the light from Southern California. It has areas where you can see through to see the stairways. It's really a classic Kahn building. Juhuli, the artist, came once to present his glass structures throughout the Institute. And here's an example of one hanging in the courtyard. One evening, I was, I think, the only person in the Institute that night. And so really got to appreciate the whole quad was filled with art from Juhuli. And this building was actually inspired, this amazing structure on the West Coast facing the Pacific was inspired by a building on our campus, which was also designed by Lou Kahn. And that's the Richards building, which doesn't have the same admiration perhaps, but is a very important architectural building designed by Lou Kahn on our campus. Jonas Salk had, of course, developed the polio vaccine, and for that, the whole country was very grateful. In the 50s, he had developed the vaccine, just like we've gone through the development of a new vaccine now on our campus. And the country, just as it was is today, was very grateful to Jonas Salk. I was fortunate to be at the Salk Institute on the 50th celebration of that polio vaccine, and we were all given T-shirts with this image of "Thank you, Dr. Salk" on them. So this was the whole institute came out to celebrate. This was the president of the institute at the time to celebrate this 50th anniversary. And I was there that day with a 6-year-old who was off school. So there is my daughter actually wearing that big T-shirt with a "Thank you, Dr. Salk" image on the front. This was my lab as I set up my lab. This was a couple of years after I started my lab, my first graduate student, my first postdoc, my first technician in the lab. This was the image of me on my ID badge from when I started at Salk. And I was there for about 14 years. I had many R01s and looked very different at the end when I left the Salk. And when I arrived at Salk, I'd never seen an R01, and it was a soft money institution. And so I was expected to have 3 R01s to run my lab. So I said to my architectural wife, they expect me to have 3 R01s and publish in the top journals. And she said, okay, that's what you'll do. So really blindly, I managed to get to the point where I had 3 R01s and also published in those journals and had a great group of people. And then it was time to bid farewell to the West Coast. And this time we headed east. And the 4 of us drove across country in an amazing journey where we saw the whole country as we came from the West Coast to the East Coast. I'd grown up in Wales. We didn't come back to Wales. This is the sign you see as you enter Wales every day, Groesa I Gymru, welcome to Wales. We didn't come to Wales, but we did come back to a place with a Welsh name, and now we live in Ballackinwood, which is a Welsh name, obviously. This is my lab at Salk saying goodbye, and this is the new lab that I managed to build here at Penn and Chop. I like to point out that all the pictures from my lab at Salk are all outside enjoying the beautiful Southern California weather. All my pictures from the lab here seem to be inside pubs drinking. So I'm here to give you a few lessons after that journey to tell you what I've learned. And so what I'm going to do today is tell you about how I went from knowing nothing about grants to being funded myself. And then when I came to Penn and Chop, to using my experience and really trying to help others do this in a more productive way. So you may ask, what are my credentials to do this? So I was fortunate over the last 6 years to be on a NIH study section, cancer etiology. So this study section met 3 times a year. I was on it for 6 years. In fact, for the last 2 years, I was the chair of the study section. And this really gives you an amazing amount of exposure. If you really want to know how the process works, go and sit on study section and get to see and read and discuss grants 3 times a year. You really come back with a sense of what makes a grant more compelling. So that gives me some credentials, but you may ask, well, can he write his own grants? All very well that he can review grants, but what does he know about writing grants? So you can go and look this up. I did this morning. NIH Reporter is a way to find out who's funded. And if you do this morning, you'll see that, not to brag, but just we've been incredibly lucky. We have 3 R01s, 2 R21s, and I'm a PI on T32 here at Penn. Okay, so I think I have the credentials, and what I want to tell you today is how we can do this as a community through a program that I started called Grant Proposal Success, or GPS. So I guess the first question we have to ask ourselves is, what is a grant? Well, it's a noun. It's something that is granted for a particular purpose. So if we're going to set out to write a grant proposal, we have to have a clear sense of what that purpose is. What is a proposal? Well, a proposal, according to Webster, is an act of putting something forward or stating something for consideration. So you will not get a grant if you don't put it forward. If you don't submit, if you don't buy a ticket, you won't win a prize. And so it's important that you go through the process and actually submit a grant that then is for consideration by others. And the more compelling that is written and presented, the better chance you're going to have to get that grant. And what is success? Well, that's what we're all searching for, the correct or desired result of an attempt. So you have to attempt a series of experiments, and you have to have some sense of what your desired result is in order to get success. Now, as Lucy pointed out, I was not successful along multiple steps in my career. So I did not do very well at high school. I struggled to get into university, did adequately at university, and also had a good time. I did a PhD in a short amount of time without the resources that American PhDs are lucky to have here. I did 2 postdocs where I wasn't sure I would make it to faculty. Eventually was very lucky to get a position. And then 11 years ago, looked around the country for where I wanted to be. And this time I was able to choose, and chose the University of Pennsylvania and the Children's Hospital. And when I came here, I didn't want to do what I'd done before, which was really write grants alone. This is how we sort of view the grant writing process, that you're locked away, keeping people away from the access, just spending times in a solitary way writing grants. And I didn't think that this was going to be the right way to do it. I'd done it that way, and I really wanted to do it in a much more open community way. And so I decided we would take a lesson from Ben. So this is Big Ben, and this is Rich Ben, the $100 note. Big Ben says, keep calm and carry on, to avoid the panic and the freak out. So that's one approach to your grant writing, is to keep calm and carry on. But I like to think that this is Benjamin Franklin. He has a slightly different approach. Ben has written his aims page, and he walks onto the campus, which he has founded, and he sits on a bench with his aims page, and he looks for inspiration. He reads over his aims page, and there's no one there to help him except for this bird who sits on the bench with him. Occasionally, especially during the winter, he manages to find someone to sit on the bench with him, although not always very helpful. And so Ben goes back to his office, and he gets together a group of his colleagues, and he presents them his aims page, and he gets feedback from the group. Now, Ben's colleagues at the time were all old white dudes, but our colleagues today are much more diverse. And he formed a club of these colleagues of his. Their purpose was to debate questions and exchange knowledge. They called these clubs Junto. The members were from different backgrounds, from different departments and occupations. They shared a spirit of inquiry, a desire to improve themselves, their community, and to help others. And this was the launching pad for many of his projects, including lending libraries and the University of Pennsylvania. So I think that this model of a junto is ideal for how we can come together as communities on this campus that he founded and work together to look at that aims page. And so the grant review communities, the GPS programs that I've set up are all on the same principle of tell me and I forget, teach me and I may remember, but involve me, participate with me, and I learn and we learn together. So everyone brings a different piece of the puzzle. Everyone has a different sense of expertise. Everyone sees something from a different corner. And when we come together and we work in groups, just like those juntos, we can put together the puzzle in a much more productive, easy, less stressful, and I think exciting, colorful way. And so this is a logo that we designed, my wife designed for the grant review communities. When these communities come together, everyone brings their different color, and it's these regions of overlap where the color really pops. And together, just like ways, we can outsmart funding. So I'm going to tell you a bit about the current state of GPS, and then I'm going to give you my philosophy on writing an aims page, which is what Benjamin Franklin wanted feedback on. So GPS are these flexible communities that are based on learning together. We share experience in grant writing. We learn about each other's science. We build communities and networks to promote collaboration all across the campus. Together, we help identify whether you're asking the right scientific question, whether you're using the right experimental approach, and do you have the right team? Who on campus may be good for you to get in your corner to assemble the best team for this project? This umbrella we call grant review communities, and it's made up of a series of GPS groups all over campus. So this GPS group started with 5 of us getting together. It was Craig Bassing, Paula Oliver, Adam Resnick, and myself, and Andrew Wells. We got together every week. We read each other's grants. We shared information that we knew about the changes that were going on at NIH and across the campus. We suggested people we could collaborate with. And then we were all very successful in getting grants. So we expanded this model first to early stage investigators and what we called ESI or Easy GPS, and then into various programs. Today, where we are is we've involved hundreds of faculty and trainees. We have intensive workshops. We have multiple faculty-led peer-to-peer GPS groups. We've done panel review meetings, grant-focused events throughout the year. We do mock study sections. We do talks like this. All of these are based on coming together to write peer-to-peer review, regular commitment to these communities, and networking as a group, both people from Penn and from CHOP. So these are all flexible community-based learning experiences where we guide each other to develop compelling proposals. We teach effective grant writing skills. And these are really cascading because these skills are going to stay with you the rest of your career. We leverage each part of the grant to maximize funding, generating realistic timelines between each other, promoting peer-to-peer review, and again, giving skills that I think are going to be passed down. It's going to become easier for our trainees to write grants when we can tell them from our successful experiences. There are all levels, so faculty and trainees. We've involved many assistant professors right across the campus. That's been the biggest group that I'd like to think that we've impacted. We have ones for faculty that are based on topics, increasing a sense of community amongst those areas. And these are really for people who want to interact in a committed way, both together as faculty or together with trainees. And they're in various topics at various stages of training. And the same, we have groups for our trainees. So we have GPS workshops that are for F and Ks right across campus. So either trainees, fellows, or people at early stage faculty. Those are over a 5-month period, an intensive workshop where we work in communities in between. And then we also do these basic science K groups where we form individual communities to support someone who's ready to submit their K, meeting a couple of times to help 1 or 2 trainees at a time. The workshop has 4 sessions. We deal with aims pages. We deal with reviewing each other's bio sketches. And then we have a mock study section at the end. We've done about 18 to 20 trainees each year, and we're now in our 6th year of this at CHOP. We take a meter of changes in knowledge and stress, and you can see we start off with a mixture of people feeling low confidence and high stress, and we really shift to where we get much more knowledge as we can track this over the course of the 4 or 5 months. I like to think that these are really neighborhoods all over campus. So they're local groups coming together in communities. Some are CHOP-based, some are Penn-based, some are CHOP and Penn-based, some are faculty-based, some are trainee-based, some are really joint. And here's just a flavor of what there is today. So from that one GPS group, 5 people coming together, we have cancer GPS, we have neuro GPS, we have a clinical GPS at CHOP, which reviews Ks from the clinicians. We have a radiology GPS, a microbiology GPS, a computational GPS. For trainees, we have a GPS workshop, a basic K where we focus on individual Ks. We just started a K2R group at CHOP. If you're on the tenure track in the med school, you're part of the Richards Society. We have an amazing Richards GPS that brings together anyone who's in the Richards Society from across the campus. That meets every Wednesday. We do once a year for those writing mirrors or DP2s, and we started at CHOP for our diversity fellows, a diversity GPS. So each of these are different types of communities at different levels of training, dealing with different topics and expertise. And this whole program has, I think, been very successful, involved hundreds of trainees and faculty across the entire campus, proven itself to be a flexible model that's different and distinct from other grant reviewing opportunities that we already had on campus, created a community culture that really buys into, I think, the culture of Penn, building morale and extending across the campus. And this is an added benefit, securing dollars in the millions across campus at the F, K, R, Mirror, and DP2 level. So you can form a new GPS. If you're out there and you feel like we're not servicing you, come see me. It could be science-focused or topic-focused. It could focus on particular types of grants, and the format is really open to you, how often you want to meet and what sort of group you want to have together. This is different from faculty reading grants. This is not a faculty reading community. This is about people coming together to read each other's grants, to help each other, to discuss in an open format, everyone in the room, just like those juntos of Benjamin Franklin, just like study section is. It's a group of faculty debating what's most compelling. We do it tied to NIH cycle. We connect people really across the campus. We help establish clear timelines, advising and giving feedback to each other. And this is all done in a peer-to-peer way at trainee level or at faculty level, with a few senior experts really helping to keep us on track. So these, I think, are some of the values of the GPS approach. It's unique. It's different from getting your neighbor to read your grant. It's really getting feedback from a whole group. These skills are going to be with you for the rest of your professional development. It's reiterative, so you can come and sit in the first round and then get feedback the second round. I think if you write better proposals, if you think more about your science, you're going to do better science itself. There are ways that we can help each other to stay on track. We've tried that we increase confidence, we decrease stress, which I think for junior faculty is really key, especially as we've seen during the last 2 years of the pandemic. And we're creating communities. They can be online communities or in-person communities. And the more money we bring in, the easier people can focus back on what they really need to be spending the time on, which is the science and research that's going on in their labs. Okay, so are you ready to start? Are you ready to join a GPS group? Most importantly, are you ready to tell your story? A proposal is like a story. It has a beginning, a middle, and an end. The beginning is where you are now. What skills do you have? What team do you have? What resources do you have? What preliminary data do you have? That's the beginning of your story. The end of the story, that's what you imagine the results are going to tell you, how they're going to impact the field. And the journey from the start to the end, that's the narrative of your proposal. So I encourage you, as you start on this, as you start as junior faculty, or even if you're senior faculty thinking about a new proposal, really think about what's your story, what's the beginning, and what's the end you'd like to imagine that you'll get to. And if you view yourself from the reviewer's perspective, this may give you some insight into those things you should be thinking about. So when you submit your review, the one chance is that the reviewer sort of likes some of it, doesn't like other parts of it, and you're going to get a very mediocre score. What you need to get is a complete thumbs up and avoid a thumbs down. You need to be in that high scoring area, that exceptional grant that has lots of great strengths and a few minor weaknesses. So you want to emphasize the strengths, minimize the weaknesses, really get to a thumbs up. So when you start on this journey, think about how compelling the reviewer would find it. They've got 10 grants in their pile. They're looking for the one that really stands out, the simplest one that they can present to their colleagues, that they can make sound compelling, that they can advocate for, that they're excited about. It's not their field necessarily. It's not something they're going to do. They may not be the expert, but they need to be excited enough that they get a full thumbs up and they can convince the rest of the panel to vote along with them. Okay, so now I'm going to focus on what I think is the most important part of that grant, the aims page that Benjamin Franklin sat on that bench and tried to get feedback from. It's just one page of this grant, but I think it's the most important for success. It's the most difficult to write, and it's the one page that everyone during the review process is probably going to read. So 3 people will have read the whole grant. They will have given a score for each of the different criteria. But the rest of the panel is going to listen to their presentation. And during those 10 to 15 minutes, they're going to go into the grant and see whether they agree or disagree with the assessment of the reviewers. And if there's discrepant reviewers, they're going to decide whether they agree with the higher score or the lower score, and they want to vote outside the range. So during these 10 minutes, they don't have time to look at the whole grant. They're really going to focus on the most important page, one page, the aims page. And that's why this page really has to do everything. It has to define the question. I have to be clear. What is it that you're going to do? I have to say, that's cool. Why don't we know the answer to that? You have to convince me now that you've raised the question that your approach is the right approach to answer that question. You have to also convince me on this page that you're the right person to do it. Not that no one else in the world can do it, but that you have everything in place that you can use that approach to answer that question. And that this will have an impact on your field. By filling that gap with this approach, you will impact your field. You will basically cross the gap, provide the critical gap in knowledge that will improve human health. Okay, so how do you do that? Well, the reviewers have to leave this page ready to advocate for you. They have to be excited for the rest of the grant. They're basically on this one page deciding whether they're going to support you or not. The reviewers have to be enthusiastic about this grant. You want them to think that it's in the top of their group of 10 or so that they have in front of them. They have to know on this page that you're going to deliver the answer to that gap. So they have to be clear about what the grant's really about. There can't be any doubt when they leave this page. And they have to know what we're going to expect, what we're going to see inside the rest of the proposal. So as you set out on this journey, ask yourself the following questions. What's already known? How can you convey what's known in the field and what your contribution is now going to be to that knowledge? What is the knowledge gap that your grant is going to attempt to fill? It could be technical, it could be conceptual. What is the overall approach that you're going to use to fill that gap? And what is the impact on the field going to be when we've acquired that knowledge? Significance to a grant is not how many people are affected by that disease. It's when you've done this proposal, when you've delivered the results that you propose, how will you have changed the field? How will you have significantly impacted the field? That's what significance is when it's being judged by reviewers. How does your background and approach position you for success in addressing these questions? So this page, as I said, it needs to be compelling. It has to introduce the big idea. It has to convey passion. You have to be excited, and the reviewer has to feel that. There has to be a logical flow. I have to understand how the aims come together to address that gap. It sets up the rest of the grant. It really tells me whether you're in the high impact area or whether you're just an average grant or whether there are significant problems because I have too much doubt. I don't think you can do it. You want to be clear, concise, if possible, memorable, and definitely succinct and easy to read. You basically want the reviewer to grab it. They have to understand the project. They have to be ready to get everyone else on board. So this is one way that I suggest you can do it, by starting by setting up the grant with 2 paragraphs. The first grant has a hook. The first line grabs the attention. Just like the first line of a book or the first line in a movie gets you excited. Go pick up your favorite novel and see what the first line is. How does it hook you? This paragraph has to state the big question, highlight the gap that you're going to fill, provide a rationale for why that's important, and explain the consequences for human health. So when I get to the end of the first paragraph, I should say, that's cool. Why does no one know this yet? This would be great if they could do it. The second paragraph then has to say how you're going to do it, what preliminary data you have, what your objectives are, what your hypothesis is, what's your overall approach? So I know, that's a cool question. And yes, that's the right approach to answer that question. I think it should have a hypothesis where possible. This is your definition of a hypothesis. Attentive assumption made in order to draw out and test its logical consequences. A hypothesis has to be falsifiable. There has to be something learned if it's wrong. We have to be testing something. A central hypothesis anchors the aims together into a common theme. It keeps the proposal focused and it has to be testable. So if your hypothesis is wrong, we're going to know something. The field is still going to be impacted. Your objectives of how you're actually going to do that. What are you going to do in this grant that will achieve filling that gap? How do they address that critical gap and the hypothesis? What do you expect them to do to impact the field? I suggest you lay out the overall hypothesis and lay out the overall objectives for this grant to address that hypothesis to fill this gap. Start with a hook that excites the reviewers, explains the importance, sets up the proposal. End the page with the impact, brings together the whole project, emphasizes the outcome, restates the gap in the team, connects the aims back to the big picture and how you will fill the gap. So this page is an iterative process. It takes time. Think about the question, think about the approach, go back and ask, do these aims really fill the gap? Do they really deliver the impact that I promised in that first line? Seek and incorporate feedback at every step. And this is such an important page. It needs ruthless editing. This is the prime real estate. Things on here need to be important, need to be clear, need to be free of jargon. So there are many things to avoid on this page. The question does not set up the grant. Often we're like, yeah, I don't really get what it's about. The approach does not answer the question. That's a great question, but that is not the approach to do it. The applicant doesn't have the experience. Great approach, great question. They just can't do it. The aims are interdependent. If aim 1 falls, aim 2 falls. Don't make domino aims. The outcomes are unclear. I don't know what they're going to learn from this. Avoid the bait and switch. The bait and switch is where you have a great introduction to your grant. You've baited them. You've set it up for one thing. They guessing what they think the grant is about. And then when they come to the aims, it seems there's a bait and switch. The aims don't seem to address the questions that were raised. Make sure that your setup is for your aims, that your aims address the question that you set out to ask. Okay, so this seems like a lot to do in one page. How do we do all that in one page? So one night, thinking about my workshop, I thought, well, we could take a rainbow anatomy of an aims page. So it needs a hook, it needs a background, it needs a gap or a hypothesis, it needs resources, it needs aims themselves, the experiments, and the expected outcome and impact of those results. So if we look at an aims page and we take this rainbow approach, we should see that we go from red towards purple, that we have a relative amount of each of these different things. So here's one of my aims pages, a grant I wrote with Susan Weiss. It has a hook. The hook is about viruses and how they maximize transcription by generating transcripts off both strands. The background and problem is that if you have strands off both sides, that you could generate double-stranded RNA. And there's the gap. We don't know whether this virus generates double-stranded RNA and what the cellular response is. Our long-term goal is to understand how viruses maximize their gene expression to prevent cellular responses. Here's some gap and hypothesis with the background setting it up. Here's the resources, the preliminary data we have. Here's how it sets up the hypothesis. These are the objectives of this grant. This proposal will identify the source of the double-stranded RNA, is it viral or host, and determine the responses activated and how they're prevented by the virus through an E3 ubiquitin ligase. You can guess what the aims are. One is probably on the source of the double-stranded RNA, and the other must be how it's prevented by the virus. So here are the two aims. They each have a short definitive title: to identify the source of the double-stranded RNA, to determine how the virus prevents the response. They each have a little bit of preliminary data. They each have a hypothesis. They each have two sub-aims. They each have a definition of what the results will tell us. There's an impact statement that retails the results and restates the resources and the team that we have to get coming together to address this. This rainbow page has gone from red through the various colors of the rainbow through to purple. I feel happy that you can take this, you can imitate it, or you can do your own type of rainbow anatomy. But think carefully about this page. For me, imitation is the sincerest form of flattery, so I don't mind if you copy this. You need a good original, and I had a great original. I am a copy of this genetic first definition, my older twin brother. And he's a great peer mentor for me. In fact, he's probably the best peer mentor I have. So find your own peer mentors and make the most of them. The rainbow also reminds us that we don't have to do things together alone, that we can do them as pairs. And in the Jewish tradition, we read throughout the year parts of the Bible, of the Torah. This last week, we read the story of Noah. Noah went into the ark to avoid the flood, taking the animals 2 by 2 in pairs, in twin pairs. And the outcome of that after the flood was the rainbow, reminding us that when we come together, we can act not alone, but in a united way, each reflecting our own individuality, but as a united community. So these are the Weitzman twins I showed you at the beginning, 1965. But these are the next set of Weitzman twins, 1999. These are my kids. I am a parent of twins, as well as being a twin myself. They were my first mentoring experiment. This was actually a school project they had, where we were determining the right amount of time to get a perfect soft-boiled egg. It was a very successful experiment. They won a prize for their poster. In fact, every kid won a prize, but they won a prize for their poster. They went on to graduate from high school, from the Jack Barrett Hebrew Academy here in Philadelphia. One went on to University of Pennsylvania, and one went on to Wash U, graduating in computer engineering and with a Bachelor of Fine Arts. And I'm very proud that they were successful at every step of their training, because they had the right mentoring to support them. And just to continue this twin theme, I would like to highlight a great podcast to help you not be a lonely pipetter. This is a podcast that comes out of Paris. The Lonely Pipette interviews scientists where they share tips, helping scientists do better science. I encourage you to go and listen to this podcast. It's really a great series. They're in season 2. These are the episodes from season 1. You may recognize some of the great scientists they got to interview. Listen to the 2 interviewers who do it, Renaud and Johnny Weitzman. Yes, that's the same Johnny Weitzman pictured here. This was our birthday that we celebrated after the pandemic in Tel Aviv this year. This is saying goodbye to my twins as they head out after college. My son actually headed west, went back to California. And there is one more set of twins. These are my twins in Israel this summer with their twin cousins. My twin brother is also a father of twins, and these are his twins born 10 years after my twins. So lessons learned. These are my lessons learned. Having started without ever seeing a grant when I started my lab, I now read over 300 Ames pages a year. So I'm happy to read your Ames page, but I encourage you, go and read Ames pages. It will make yours better. I've learned since coming to Penn, we have an amazing community of researchers across the Penn and CHOP campus and a really generous faculty who are always willing to give of their time. So really use them. They're an amazing resource. And I learned that success comes from hard work, persistence, and although a lot of failure, if you focus, you can get success. And my lessons to you are: make your proposal compelling, succinct, clear, and exciting. Good proposal preparation will make your science even better. You'll know what the experiments are that you should be doing. You'll know what the expected results are, and you'll know if your hypothesis is wrong, how they'll impact the field. Writing grant proposals does not have to be a lonely pursuit. Do it as part of a community. Use these community of peers to help you get to grant proposal success. It's where the different parts of our community come together that the colors really pop, and we can outsmart funding together and have great success. What is success? Success is not the key to happiness. Happiness is the key to success. So love what you're doing and you'll be successful. And it's clear from this morning's light on our buildings that there's a bright future for faculty at Penn and CHOP. And with that, I will stop sharing and happy to take questions.
Thank you so much, Matt. We have 10 minutes for questions. You can just put your name in the chat or you can raise your hand. We would love to hear from you. Carmen.
Thank you, Matt. This is not only a wonderful endeavor, but you tell it in such an inspiring way that I really do think you have a special gift for communicating this in a way that we need to hear it more. I have two questions. We're at Focus, and so I was wondering the proportion of women and underrepresented minorities that are engaged in this GPS program. And the second question is, what are the challenges to scaling this throughout our entire school? Clearly, it's a gem of a program, and I'd love to see it scaled, especially at PISA. Thank you.
Okay, great question. So I'm still learning about storytelling. I actually went, if anyone listens to the Moth podcast, I actually went to a Moth live storytelling this week, and I was so scared at the prospect of standing up in front of an audience and telling a story. It's much easier to give a seminar, I think. But I think storytelling is really, it is key. It starts with a beginning, has an end, and you have to know a good, compelling narrative that keeps your audience engaged for the whole time. That's really what proposal writing is. Good question about who are we impacting? So we started as a small group of 5. We expanded to early stage investigators. We helped people write their first Ks. So the first successful K through basic GPS was actually Abby Green, who was a fellow in my lab, who now is an assistant professor at WashU. And then the med school asked me to come and talk to them, and they said, this sounds great, but we can't do it. And I said, why can't you do it? And they said that we don't have anyone to drive it. And so I said, well, I'll drive it. So we started working with the Richard Society. I would say that all our programs, that's a good question. We definitely have more female participation than male. I don't know whether that's reflecting generally what our CHOP or more CHOP focused faculty looks like. I am learning a lot from the process about how to engage and appeal to different audiences. It's not all pediatrics. I did a GPS for K to R with clinical psychologists. That was a real eye-opener for me. I would say the Richard Society GPS that Kate Hamilton and Mary Mullins and I, together with others, have been running has been one of the most fun. Kate Hamilton once and I were walking back when we did it in person, and she said, this Rich's GPS is the best hour of my week. And I said, absolutely, it's the best hour of my week too. So I know probably more than most about what science is going on campus, because I've just been exposed to every junior faculty who comes through. I really have a great sense of who's doing what, and it allows me to connect people beyond GPS. Diversity. So we started a Diversity Fellows GPS here at Penn, at Job. And again, I think I'm learning from this group. I'm the only old white dude in this group. And I'm really getting them to interact and support each other. I'm learning a lot from the process in my own education of how to deal with DEI and different communities. And I've learned a lot about different science. So my science, most people don't do science like I do, which is very molecular and cellular. And so it's been great to realize that you can get a radiologist and a biochemist in the room, and they can both help each other. And if they can both understand each other's grants, they're going to have a better time out there. Scalability. So we started with 5 of us. We've scaled to, I would say we've probably impacted 300-400 now. This was all pretty much with no resources, led by me and a few generous individuals. Now we're starting to set up structure. So in the next year, I think we're going to see many of these across campus, and I hope they'll be available to anyone who's interested. Some will go, some will come, depending on what's needed at the time. Different fields are different in their sort of frequency of submitting grants. Different people need different things at different times. There's not one. It's not formulaic. It's everyone's writing an individual grant. But I'm looking for partners. So anyone wants to reach out to me. I love working with new groups, and I'm also going to other institutions around the country. I don't know why it should just be CHOP. We did a webinar where we had 400 people, which was Wistar, Penn, and CHOP, and other institutes in the country, in the city, and the people from outside of Philadelphia also joined. So I think we can make this across the country.
Others?
Matt, I had a question. Awesome as always. My best name pages have been attributed. I attribute them to you. So in our small group, any tips for the approach? That's where I always get slaughtered. I'll admit it.
Okay, so we focus on the Ames page just because it's really hard to write. But you know what? In our clinical GPS, we spend a lot of time helping people on Ames pages, and then we saw some of the rest of the grant. And those of us who are running it are like, whoa, we thought we were teaching skills that would like translate. So I would say take the skills and the emphasis that you put and the clarity of your Ames page and transfer that over to your grant. The second thing I would say is, I learned this from one of our GPSs. I really aim for symmetry in grants. So if I have 3 pages describing aim 1, I have 3 pages describing aim 2. If aim 1 has a rationale and then 2 sub-aims, aim 2 has a rationale, and the reader knows where they're going to expect to see something. They're like getting used to the flow of your grant. So I think flow is really important. I'm a little bit obsessed in symmetry now. Perhaps it's being influenced by an architect. So I think you should have the similar ruthless editing, and then figures. Figures are your way of conveying that there is feasibility. They're not the same as figures for your paper, which go A through K or J or L or Z. They're selective pieces of data that demonstrate that you have feasibility. They have a legend that describes the grant so that I completely understand what the data are that you're presenting, that describes your statistics. Basically, they convey rigor, feasibility. They tell me what type of scientist you are. So I hate it when I don't know what the experiment is, because I look at the legend and it doesn't tell me. In fact, I can kill a grant by saying, I don't know about this grant. I wasn't the reviewer, but figure 17D, is that a Western or a Northern? Now everyone on the panel is like, there are 17 figures. They go to figure 17D and they blow it up and they don't know, are they looking at RNA or protein? So that's undermined rigor of that grant, because they haven't conveyed in their own data how rigorous they are and how clear they are with presentation.
Thanks, that was great.
Others? Any other questions? I did want to say that we've recorded this session, so when Matt says imitation is allowed and celebrated, he really means it, because this will be available to people, because there's a lot of information that people can use quietly as they're struggling to do their AIMS page. So if there are no more questions, I just want to say, Matt, that was the most compelling and interesting discussion of research and how to do it that I've ever heard. So I really appreciate it. And I love your authenticity about the ups and downs of a career. So thank you, thank you, thank you. And if people want to talk to you, I take it they can contact you, that you're open to that.
Just reach out. We can talk over Zoom, or we can have coffee together. And I want to thank you for what Focus does. This is helping women on campus. It's also helping everyone on campus coming together as a community to share honestly what we're struggling with. And I think that this is just such a perfect forum, and will help give me focus too.
Thank you so much, Matt.